Hello, hi, wishing you a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my class. So today we are talking about lecture 4 of biomolecules. So here we are going to finish this chapter. So we are going to talk about enzymes in detail today. So this will be the final lecture of your biomolecules. Hi Prince, yes, a very happy good evening. Thank you Prince for joining my lecture today. So please do like, share and subscribe. Yes, Prince, so why didn't you turn up for my 4 o'clock lecture? So we were discussing the previous year question. That's fine. Okay, so I hope you have shared the link. So Prince, please do like, share and subscribe. Come on, quickly share the link with your friends and then please join me for the lecture. So I will try to finish off enzymes fully because I have given you an introduction to what is metabolism, what is a metabolite, what is metabolic flux. So we will move on from there. Okay, yes. So I will finish it off today. So tomorrow's lecture, tomorrow's schedule, so this is lecture 7 of Mendelian genetics. So tomorrow we'll talk about your genetic disorders, what are chromosomal disorders, what are Mendelian disorders. So Mendelian disorders are hereditary, so we will talk about them. So your concepts like your color blindness. So your concepts like color blindness, sickle cell anemia, so all that and problems based on that we will discuss tomorrow. Your hemophilia. So all that we will discuss in tomorrow's lecture, that is lecture 7. So please do join me. So I will also teach you what is pedigree analysis, or I will teach this separately in lecture 8. Okay, and tomorrow at 6 p.m. So today I'll finish this chapter. So tomorrow we are discussing important questions from biomolecules. Okay, so this is my schedule for tomorrow. So we are in lecture 4 of biomolecules. So moving further. So giving you a quick recap what we read in the previous lecture. So in the previous lecture, we spoke about the nature of bonds. So the previous lecture happened yesterday at 6 p.m. Hi Vyrovan, yes, after a very long time you're here. Yes, a very happy good evening. I'm good, thank you. How are you? So thank you so much for joining my lecture. So please do like, share and subscribe. Hi Shimpi, yes, a very happy good evening. I'm good, how are you? So thank you Shimpi for joining my lecture. So please do like, share and subscribe. Okay, so nature of bonds. So between two amino acids, so you have hydrolysis reaction happening. So thus it leads to the formation of your peptide bond. So this is between two amino acids, right? Then in between two monosaccharides, how it is attached? Okay, Vyrovan, okay, Shimpi. So between two glucose or between two monosaccharides in a polysaccharide, so we again have hydrolysis reaction and we have the formation of glycosidic bonds. Right? So hi Keshav, I heard you're busy but you're back. So thank you so much Keshav for joining my lecture. So please do like, share and subscribe. So and in between in your nucleic acids, if you're talking, so your nitrogenous base is attached to the sugar via your glycosidic bond. Your sugar is attached to the phosphate moiety by your phosphoester bond. Okay, so this is in your nucleic acid. Then we spoke about the structure of DNA. What is the structure of bDNA, right? So we spoke about the structure of protein, the primary structure, the secondary structure, etc. Pavitra, hello, hi. So thank you for joining my lecture. So please do like, share and subscribe. When will you take NCERT line by line decoding series? So that I am not taking because it was already done once. So I'm not doing the repetition. But if you have doubt in any particular line in NCERT which you do not understand, yes, you can ask me. Okay, yes. So then I spoke to you about the structure of DNA, how DNA is formed, etc. So then we went on to talk about what is metabolism and how biomolecules are measured. How they are measured? So they are measured as moles per cell or moles per liter, right? So that is how it is measured. So further, we spoke about something else also. That is, what is a catabolic reaction? What is an anabolic reaction? So when I talk about metabolism, so metabolism is the sum of all chemical reactions that is happening in your body, right? So metabolism can be of two types. So that is catabolism and the other one is anabolism. Yes? Okay. So this catabolism is when you have larger molecules. When it is broken down to form smaller molecules, it is catabolism. It is like climbing down a stairs. So when you climb down stairs, you release energy. So our body needs this energy and it traps them in the form of high energy bonds, that is as ATP. Okay, yes. What is the difference between normal and systemic anatomy? Okay, where do I get line by line decoding lecture? Okay, so in your eCareerPoint NEET channel, that is in Hindi medium, so Avneet ma'am has done decoding NCERT. So you can look into that. Okay, Pavitra? Yes? Okay. So Keshav, what is the context in which you saw these two terms? Normal and systemic anatomy. So what is the context you saw these two terms? So let me know. Yes? Yes, ma'am, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay. You want an English lecture, ma'am? Okay, so if maybe I can take it for 2022 NEET, but not for 2021 NEET, so that I have a very, very short time. So that is the problem. Okay, Pavitra? Yes? Okay, so this is catabolism. Anabolism is from small molecules, you form larger molecules. So like you have the formation of cholesterol from your acetic acid. So this is cholesterol. So this is anabolic reaction. So this is also known as synthetic reactions. So this is like climbing up the stairs. So here you have to supply energy. So these are the two reactions of your metabolism. Clear? Okay? Fine. So this is a quick recap of what we read in the previous lecture. So the living state now. Ma'am, one MBBS student from Russia told this in that they teach systemic anatomy, but in Indian anatomy, they teach in different way. That's why I asked. Okay, so systemic anatomy means how the body is functioning. Like systemic anatomy is where you have combination of two or more parts together, like how your endocrine system works along with your nervous system. So normal anatomy is when you read each one separately. The structure of neuron, the structure of nervous system, that is separate. Then the structure of muscle, you read it separate. So this is normal anatomy and this is systemic anatomy. Okay, Keshav? Yes. Hello, Kathy. Yes. How are you doing? I hope you're fine. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay. So moving from here, we are talking about metabolism. So moving further from here, metabolites or biomolecules present at a particular concentration in your living system. So you have a lot of biomolecules, right? So you have your glucose molecules, which are your carbohydrates. You have your proteins, you have your hormones, you have sugars. So all these are basics are biomolecules, right? The base is a biomolecule because they all contain carbon containing molecules. But what is the major difference? The concentration differs. So glucose is needed in high concentration in your blood, whereas your hormone is needed in very less quantity. A hormone should not be high. So when a hormone is produced in excess quantity, we call it as hypercondition, hyperthyroidism, hypocorticoids, right? So that is the condition. So what is characteristic to a biomolecule or a metabolite is that it should be present in a particular character. So example, blood concentration of glucose in a normal healthy individual should be from 0.2 to 6.1 millimole per liter, and hormones should be in nanograms per liter only. So the concentration should not get inverse. Yes, Dharani, a very, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So in fact, Biological systems, all living organisms, exist in a steady state characterized by concentration of each of these biomolecules. So biomolecules have a metabolic flux. So when I need hormones, it is produced in high amount, but always hormones are not present in high amount, right? So that is one thing. Suppose if I want antibodies. So right now, touch wood, we are not suffering from any disease, so our body will produce less amount of antibody, right? So there's a constant state or change in the quantity of these biomolecules. So this is my metabolic flux. So any chemical or physical process that spontaneously moves to equilibrium. So steady state is non-equilibrium state, that is the living state that able to perform work. So in your physics or chemistry, yes, so what is equilibrium? So equilibrium is when it spontaneously moves to an equal quantity, equal entity, right? So that is equilibrium. Suppose if I have two cells, for example, so let's go here. Suppose I have two cells, one is cell A, one is cell B. If the concentration of ions here is high and the concentration of ions here is also high, then these two are equal. Right? So we call this as equilibrium condition. Right? But in a living system, this cannot be possible. Why? So if everything is equal in our body, for example, if the concentration of antibodies, if the concentration of WBCs, if the concentration of hormones is equal all throughout our body, then it is called as the state of death. It is not the state of equilibrium. Okay, yes sir, good evening. So thank you so much for joining our session. So please do like, share and subscribe. So why so? So there has to be a difference in concentration because your metabolites are dynamic molecules and you have what is known as a metabolic flux. Right? So it should not be same throughout your body. So equilibrium in a living state is non-steady state. So you can call it like this. So steady state in a living system is non-equilibrium state. So whereas in chemistry, so what is steady state? So here the steady state is your equilibrium state, right? But in the living system, the steady state is your non-equilibrium state. If you have equilibrium in your human body, it means the person is dead. So in that case, if a person has to continue working, so we should make sure that he is in a non-equilibrium state. Okay? Yes? Okay, so Sanjeevani, hi, hello, a very, very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Ma'am, if I start now, can I complete NEET preparation? So are you a beginner or do you have a base? So that matters. If you have a base, then yes, constant and consistent determination, you can definitely cover your syllabus. Okay, Sanjeevani? Yes? Okay, so I hope everyone has understood what is steady state equilibrium and non-steady state equilibrium here. Yes, so in a chemical equation, steady state is your equilibrium reaction. In a living organism, steady state is a non-equilibrium state. Okay, yes, done? Fine, so this is what is mentioned in my slides here. So now we are going to talk about something most important thing in a living organism, without which a living organism can never proceed forward. Be it any living organism, be it a bacteria, be it an animal, be it a human, be it a dinosaur. So without enzymes, yes, you cannot survive. So enzyme is an important characteristic of a living organism. If a person has to be in a non-equilibrium state to maintain a steady state, then he has to have enzymes. So without enzymes, he has a lot of physical disorders. If there is a mutation in a particular gene that is coding for an enzyme, then that is called as a genetic disorder, right? For example, phenylketonuria. So enzymes play a very important role. Even the simplest of reactions in our body are catalyzed by enzyme. Why? What is the use? So when I can do it without the help of enzyme, for example, enzyme is a catalyst, right? So suppose I have a reaction. So A gives me B. Okay, so in the presence of an enzyme E1. So even without E1, A gives me B. That is nature. Okay, but why should we have an enzyme? So without the absence of enzyme or without the enzyme, A can give me only 3 molecules of B per second. This is my hypothetical example. Okay, but with my enzyme, A can give me 3 million molecules of B per second. So that is the efficiency of enzymes. Okay, so if you want to talk to me, yes, you can come to quota and you can talk to me, but the more easiest way is you can ping me in the live session, you can talk to me in the Telegram doubt session. Yes, so these are various ways you can meet me in the online class. So this enhances your time, this saves your energy, etc. So it's the case with your enzyme. So that is why enzymes are absolutely essential, and your human body slows down without enzymes. Okay, so now in the present case, for you to come to quota all the way is really tough, right? So similarly, for A to produce B without enzyme is really tough, and our body needs so many million molecules. So what will your body do with just 3 molecules, right? So that is the reason enzymes are absolutely essential. So what are we going to talk in your enzymes? So under the topic enzymes, we'll compare it with your chemical reactions. Yes. How does it bring about high rates of conversion? Nature of enzyme action. So how does your enzyme actually work? How does it proceed to give you a product? Then factors affecting enzyme activity, classification and nomenclature of enzymes. So that is the final topic. So these are the 5 topics that I'm going to discuss now. So please pay attention. So enzymes are pretty important. So if at all you get a question, so you should not get struck. So enzymes are proteins. So you have the saying. So all enzymes are proteins. All proteins are not enzymes. So all enzymes, irrespective of whichever enzyme it is a protein. Okay, but all proteins cannot be enzymes because some proteins are antibodies, some proteins are hormones, some are normal transmembrane proteins. So that is the reason. Okay, so please remember these 2 cliches, it is very important. So all enzymes are proteins. So some nucleic acids behave like enzyme. One example is your RNA. So your RNA has catalytic activity because of the 2' OH it has in his ribose sugar. So that is the reason it has a catalytic activity, and your RNA functioning as an enzyme, it is known as your ribozyme. Okay, only some nucleic acids can behave as your enzymes. Okay, clear? Only few exceptions. Yes, you're absolutely right, Keshav. Okay. Hi, Antonia B. Yes, a very happy good evening. Yes, Shubham sir, good evening. So thank you so much both of you for joining my lecture today. So please do like, share and subscribe. So that is your ribozymes. So an active site of an enzyme is a crevice or pocket into which the substrate fits. So how does it help? So enzyme helps function in quick reaction. Quick, and also it gives you a large number of products. So that is why you use an enzyme. Okay, Shimpi? Yes. Okay. So, for example, if I have an enzyme molecule, if this is my enzyme, so I have my substrate, this is product. So, suppose if my substrate is A and my product is B, my enzyme is E1. So, how does this happen? So, my substrate binds to the active site of the enzyme. So, the site or where your substrate can bind in your enzyme, that is known as your active site of an enzyme. So, your substrate will bind to the active site of an enzyme and then give you the product. So, you should remember this term, what is an active site. So, this is important. Okay? Yes. Suppose this is my substrate. So, maltose is my substrate. So, maltose is a disaccharide. It has 2 monosaccharides. So, this is my enzyme. So, assume my enzyme is E1. Okay. So now this substrate will bind to the enzyme at a particular site known as active site. So it is exactly perfect. So your maltose molecule exactly fits here. So understand from this diagram that each substrate has a specific enzyme, or an enzyme is specific to a particular substrate. So the same enzyme will not react with sucrose. The same enzyme will not react with galactose. Okay, so this enzyme is particular for each substrate. So you have n number of enzymes, you have n number of substrates in your body. Clear? Yes. So your substrate will fit perfectly into your active site, and it is further broken down into two glucose molecules. So your maltose is a disaccharide consisting of individual monosaccharides called as glucose. So now these two become the product and your enzyme comes off. Okay. Yes. So your enzyme has to remove from the reaction, because ultimately we need only the product. We do not need the enzyme, right? We need the enzyme to produce the product. So this is how it works. Ma'am, these enzymes are exocrine or endocrine. If it's endocrine, then it is hormone, right? So these enzymes are produced by your cell. So enzymes are proteins. For example, if I need a particular enzyme known as maltase, if I need an enzyme known as lipase, so these are present in my pancreatic juice. Okay, so depending on the function, it is present. So pancreatic juice produces these two enzymes. Suppose if I want to produce a particular enzyme, that is phenylalanine hydroxylase. So phenylalanine is an amino acid. So phenylalanine has to be converted to tyrosine in your body. So for this we need a particular enzyme, phenylalanine hydroxylase, that is PAH. So where is this enzyme produced from? Where is this enzyme produced from? So this enzyme is coded by a particular gene. So this gene has to first be replicated, then it is transcribed onto the mRNA, and finally it produces this protein, which further takes its position as this enzyme for this particular reaction. So depending upon the particular need, it is produced in particular locations. Okay, Kesha? Yes. But all proteins, all enzymes are produced like this. So if I want a particular enzyme known as dehydrogenase, yes, I have to have an enzyme or a gene that is coding for it. So if I want to have an enzyme trypsin, so trypsin is present in your inactive form in your pancreatic juice. So depending on the function, it varies. Okay? Yes. Ending with ASE, RR enzymes, but trypsin is also an enzyme. So you have exceptions also. Okay, Vishal? Yes? Means it releases into the duct. Okay, if it releases into the duct, then it is exocrine. So they can be enzymes. Yes, okay. So for example, even bile has a duct, but bile is not an enzyme. Bile is a substance which has enzyme. No, bile has no enzyme, but it has two pigments, right? So it's not specifically duct based on ducts. So I hope you have understood how your enzyme works. Yes. So moving further. Okay. So what is the function, or what are the important features of your enzyme? So enzymes are remarkable because you cannot produce, or there's nothing which can enhance the rate of the reaction like how your enzymes does. Your enzymes are highly specialized protein. They know which enzyme will catalyze which reaction. Okay. So next one, bilirubin and biliruridin are the pigments of bile. Yes, absolutely right. Then they have extraordinary catalytic power. It's not that a normal reaction produces 10 moles of B and an enzyme catalyst reaction produces 12 moles. No, it is not like that. It produces million times more. So it has extraordinary catalytic power. It has high degree of specificity for a substrate. So you have n number of enzymes for n number of substrate. How many ever substrates you have so many enzymes. Let's for example take glycolysis. So glycolysis is your 10-step process. So what is the first step? So glucose gets converted into glucose 6-phosphate. The enzyme is hexokinase. So hexokinase will not bind to maltose. Hexokinase will not bind to fructose. So hexokinase is very specific for your glucose, so that it gives the product G6P. That is glucose 6-phosphate. It has a high degree of specificity for the substrate. So it accelerates the chemical reaction tremendously. If A to B, it takes 2 minutes, but it produces millions of molecules in a second. So it consumes or it saves less time. Okay. So found in aqueous condition, that is mild pH and temperature. So enzymes basically are proteins, so they have an ability to denature. So they work in aqueous condition, that is they work in mild pH and temperature. So your body temperature is 37 degrees Celsius. So if I go to a cold spring where the temperature is around 110 degree, so my enzymes will not work because they are basically proteins and they tend to denature. Yes, clear? So this is about the features of the enzymes. So moving further, so now, so I guess it's a repetition slide, okay. So chemical reaction. So physical process and a chemical reaction. For example, when I say, so ice melts to water. So ice is solid and water is liquid. So here I have a change in state of matter, right? So this is your normal chemical reaction that happens. For example, when your starch is hydrolyzed to glucose, so this is also a chemical reaction, but here the state of matter is the same. There is no much difference. So the rate of physical or chemical process, so what is the rate of the reaction? So this reaction also has a rate, this reaction also has a rate. So what is the rate of this reaction? So the rate of physical or chemical process refers to the amount of product formed per unit time. So I have defined what is rate when we were discussing about plant growth and development, right? So rate is increased growth per unit time. So similarly here rate of the reaction is amount of product formed per unit time. Yes, the same thing here also I am saying. So more the temperature, the enzymes will not be able to work. Okay, Antonia B? Yes. So the rate is very, very important. That defines the efficiency. So this is a very important condition. So please pay attention. Rate doubles or decreases by half for every 10 degree change in either direction. Suppose our body temperature is 37 degree. Okay. So if I move to a temperature where it is 47 degree, or if I move to a temperature where it is 27 degree, the rate of the reaction can either decrease or it can double by half for every 10 degree rise in temperature in either direction. Okay. So this is the simplest equation which we have read in your breathing and exchange of gas. So what, okay. Yes, so water plus carbon dioxide is your carbonic acid, right? So this carbonic acid helps in the attachment of your carbon dioxide so that it can be transported. For this you need an enzyme known as carbonic anhydrase, right? So if you have this enzyme, so this is the simplest reaction. I am just giving you an example. So it is reversible in both the direction. So remember this much. So the power of enzyme is incredible. Why? Because for this you need an enzyme carbonic anhydrase to produce your carbonic acid, right? So you need an enzyme known as carbonic anhydrase. So this carbonic anhydrase, if it is present, okay, so it will give me around 200 to 300 million molecules of carbonic acid if it is present. Otherwise, without this enzyme, my cell forms pretty less molecules of carbonic anhydrase. Okay. So the power of enzymes is incredible indeed. So thousands of enzymes, each catalyzing unique metabolic reactions are present. You have multi-step chemical reactions each step is catalyzed by the same enzyme complex or different enzyme in a different metabolic pathway. So for example, if you talk about link reaction in plant physiology, so where your pyruvic acid gets converted into acetyl-CoA, you have your pyruvate dehydrogenase complex. This pyruvate dehydrogenase is a multi-enzyme subunit. It has 5 subunits if you remember. So some things like that, so that is mentioned here. Okay. So glucose getting converted to pyruvic acid. So this is the process of glycolysis. So this is a step in your metabolic reaction. So this is 10-step catalyzed reaction. So how does this conversion happen? How does your enzyme work with the substrate? So imagine I have a substrate. So here my substrate is A. So it is getting converted to the product B. And here my enzyme is E1. So how does it happen? Yes, 4 cofactors and 1 Mg2+, right? So 1 ion, metal ion we have. Yes. So how does this happen? How does this rate of conversion takes place? So imagine, so now you know what is an active site. Suppose this is my enzyme. Okay. So I am just drawing, this is my own imagination. This is my enzyme. So I have a substrate. So my substrate is A. Okay. So initially the substrate will come near the enzyme or the enzyme near the substrate. So then what happened? Your enzyme and your substrate will bind together. So like this. So this is my substrate. So your enzyme and your substrate has bound together. But this binding is for only a short period. So if I put it in bracket, it means it is only for a transient period. Okay. So now slowly this will become enzyme product complex. So the enzyme is converting the substrate. So it will quickly become enzyme product. And slowly your enzyme will be released and your product will be released here. So this is how the conversion happens. Is this clear? Yes, how the conversion happens? Yes, any doubt? So this is the simplest way how does this conversion happen. Yes, clear to everyone? No doubts? Guys, come on, please do like the session. So today you have forgotten to like it, so please like the session. So this is clear? So the same thing, whatever I taught you is written here. So you have an active site, it forms enzyme substrate complex, then the products comes out, then the enzyme comes out. Okay? Okay. So now the most important thing, the concept of activation energy. So what happens? So don't get confused. So from this enzyme will be consumed. So what is your, so now what will happen to this enzyme? Is that your question? Okay, so now this enzyme has done its work. So either it will be degraded or it will go for the next round of reaction. So there are some products like your glucose getting converted into glucose 6-phosphate has to happen continuously. So it will be ready for the next round or it will be degraded by your degradative protein or your ubiquitin complex. Ubiquitin is a family of protein which destroys unwanted proteins or enzymes in your body. Fine? Okay. So now what is this concept of activation energy? So I have taught you what is active state, how the enzyme happens, how it binds, etc. So our only confusion is what is this activation energy? So we write it as EA. So look into this graph. So whenever a graph is given, don't panic. So first thing you should do when a graph is given is read your x-axis and read your y-axis. So x-axis is the progress of a reaction. Suppose if I have a reaction A giving B, so what is the rate of progress? How quickly it is getting formed? So this is the potential energy that is invested to get it converted into the product. Okay, so now look, I have two peaks, one in blue and one in red. So look at the one in red. So this is my peak in red. So this says, so this is my substrate concentration. Okay, so here I have maximum time. So here the progress takes much longer to form the transition state. So what is transition state? So your enzyme binding with substrate is the transition state, and this transition state is longer when there is no enzyme. So meaning when there is no enzyme, for the conversion of B takes much longer. But when there is enzyme, this transient period is very short and it quickly gets converted into product. Okay, so initially when I have an enzyme and when I have a substrate, so this concentration of substrate plus the substrate concentration when it has bound to the enzyme are two different things, because here it is transient, it is on the verge of getting converted to product. So the difference in the energy for the formation of substrate here, or the difference in the energy of the substrate here and the difference in the energy of substrate when it is in the transient state is what is your activation energy. Okay. So if you have more activation energy, so you need more time to form the product. So it is like this. So more activation energy, so then you need more time. So less activation energy, less time. So your transient ES complex decreases the activation energy. Clear? So you understood how activation energy is related to the rate of a reaction? Yes, everyone, is this clear? Yes, any doubts? This is clear to everyone? So lesser is the activation energy, more quicker your product will be formed. Yes, okay. Any doubt? Any doubt? Can I move forward, everyone? Okay, so Shimpi says repeated. I am sorry, Shimpi, I didn't see. So which one you want me to repeat? I am sorry, Shimpi, I didn't note. So this one you want me to explain? Yes. So Shimpi, see, I have an enzyme, I have a substrate. So the enzyme has an active site, right? So the substrate binds to the enzyme, but the substrate binds to the enzyme only for a short time. So when I put it in bracket, it is for a transient period, because it will quickly change into enzyme product complex. Now your enzyme will come out and your product will be formed. Clear, Shimpi? Yes? So the concept of activation energy is clear to everyone? Okay. So what is the nature of enzyme action? First it binds to the substrate, then it forms your enzyme substrate complex. So all that is written in theory, but I have explained it to you with my own writing. So the same thing is written. So when you download my PPT, you can go through them. So now what are the factors affecting enzyme activity? Suppose in a particular person, right? So say for example a person P is suffering from excess activity of an enzyme. So he has excess production of enzyme. So his cell has no regulation of enzyme. So just like when you have excess hormone, it is an issue, you should not have excess enzyme. Suppose if this enzyme is performing this function, so at one point you will not need B. So unnecessarily you are wasting energy, right? So how in this case you will alter the enzyme activity? How will you stop? So for this cases you should know what affects the enzyme activity, so that you can stop this excess activity of the enzyme. So activity of an enzyme is affected by a change in conditions that can alter the tertiary structure of the protein. So temperature, pH, change in substrate concentration or binding of the specific chemicals can regulate its activity. So my body temperature is 37. So if I go to a temperature of 67 degrees, my enzymes cannot perform. So if I draw a graph like this, yes, and if I draw enzyme activity, if I draw temperature, so my enzyme will show maximum activity at only one particular temperature, right? So this is known as optimum temperature for the activity of enzyme, and so is in case of pH. So if I have an enzyme in my stomach, it will work in a pH of 1.8 to 2 only. If I have an enzyme in my intestine, it will work only in alkaline pH. So pH is also an important concentration. Okay. So change in substrate concentration. So what is this change in substrate concentration? So suppose I have my enzyme E1. So this is the active site of my enzyme. So I also told you every enzyme is specific for a substance. Is specific for a particular substance, right? So now I have the substrate A. So this is my substrate. But I have a competitor to this which is also similar in shape. So let me draw it in a different color. But this is C. So this is also similar to A in structure. Now, always the bad things have more affinity, right? So if I give you a textbook of plant kingdom or if I give you a movie, so all of us will go for the movie, right? Because that is a very good entertainment and bad things attract us more. So similarly, just like your hemoglobin has more affinity for your carbon monoxide than for oxygen, your enzyme always has more affinity for the competitive substrate. Okay. Ideally, only this should bind, but this has more affinity or this is more in concentration. Okay. So it is not that for one substrate you will have one enzyme. Suppose if I have 100 molecules of A, I will have 100 molecules of E1. Okay, understand like that. So if I have 100 molecules of A, if suppose I have 200 molecules of C, so C will quickly bind to the enzyme. So now instead of A binding, my C will bind here. If C binds to E1, then the process will not take place because the enzyme is specific to a particular substrate. Though C mimics the structure of A, it cannot carry forward the reaction. So this is competitive inhibition. Okay, yes. So this type of happening, so this is the inhibition reaction. Inhibition is to stop. So what type of inhibition? Competitive inhibition, because the structure is same. So this process is competitive inhibition and this molecule is known as inhibitor. Clear everyone? Yes? Okay, so Vaishali, yes, a very happy good evening. So thank you so much for joining my lecture. So guys, please do like, share and subscribe. So here I am talking about biomolecules. Is this clear? So I have this particular enzyme, succinate dehydrogenase and malonate, right? So that has a competitive inhibition activity because two substrates mimic that. So succinate dehydrogenase is an enzyme. So that has two similar substrate which acts or which gives competitive inhibition. Okay, so please remember these cases. So I guess I have the graph. Yes, pH, temperature. Okay, so I explained what is competitive inhibition. Ma'am, I didn't find your PK playlist. So what is that PK playlist? I don't know what is it. Okay. So now what is this graph? So what is this graph? So I told you the activity of enzyme is affected by temperature, is affected by pH, is affected by the substrate concentration as well. How? How it is affected by substrate concentration? Okay, plant kingdom playlist. Okay, I will send it to you, Keshav. Okay. So how the concentration of substrate differs? Increase in substrate concentration increases the velocity of enzyme reaction. So pay attention. So if suppose I have 50 substrate molecules and I have only 30 enzyme molecules. So now quickly all the substrates will bind to the enzyme. So all the 30 substrate will bind to the 30 enzyme and no enzyme is free. Right? Now the activity of enzyme will be at its peak. The velocity of the enzyme will be at its peak. This maximum velocity with which an enzymatic reaction can attain is known as Vmax. Okay? Yes? Fine. So, now this is maximum velocity is Vmax. But the concentration of substrate taken to reach half Vmax is Vmax by 2. So, I will go back, I will explain here. So, see, I have substrate concentration, I have velocity of reaction. Okay. So, now as my substrate concentration increases, the velocity of the reaction reaches the maximum. Right? This is Vmax. Then, what is the concentration of the substrate that is needed to reach half the maximum velocity? If this is 100% velocity, and how much substrate do I need for 50%? So, that is Vmax by 2. So, for this, I plot a line in the graph and I find the substrate concentration. So, the half maximal substrate concentration needed to reach half maximal velocity is my Km value. So, Km value gives the concentration of substrate that is needed to have half velocity, half the maximal velocity. Okay? Yes? So, this is Km. So, this is a constant. So, this is known as Michaelis-Menten's constant. So, he was a scientist. Now, clear? So, you understood what is Vmax? Vmax by 2. What is Km? Yes, any doubt? Okay, so I will share the link with you. So, please send me a reminder. I will send it to you. Okay. So, this is clear? Yes, everyone? No doubt? Okay. So, this is concentration of substrate. Fine. So, now I have to teach you last two things, classification of enzymes. So, here I am going to teach you only limited classifications, not all the classifications. The first classification of your enzyme is oxidoreductases. So, this catalyzes oxidation reduction reaction or dehydrogenation reaction, or sorry, dehydration, or you can also call this dehydrogenases. So, this is the first type. When an enzyme catalyzes oxygen reduction reaction, it is known as oxidoreductases. So, it is between two substrates. Suppose I have a substrate S. Okay, so this is oxidized and I have another substrate S dash, it is reduced. So, now if I have an enzyme that is oxidoreductase, so I will have S reduced and I will have S prime oxidized. So, this is oxidoreductase. Okay? Yes? So, number 2, transferases. So, it transfers a particular group between two substrates. So, I have two substrates S and S prime. So, S has a group G plus I have a substrate S prime. So, the answer for this enzyme, that is transferase enzyme, will be S prime G plus S. So, this is transferases. Okay? So, Vaishali is also asking for biological classification. Okay, so you guys tell me one thing honestly. So, should I teach biological classification? So, do you still feel biological classification tough, or do you want me to watch, do you just want me to send the video to you? Yes? So, should I teach biological classification? Is it still tough? So, should I give you a recap of biological classification? So, let me know right now. Because biological classification, time and again, you people are not satisfied. So, should I teach you once again? Yes, let me know. Okay, so this is transferases. Now, number 3, what is the third type? This is hydrolases. Hydrolases, it causes the hydrolysis of the bonds like your ether bond, ester bond, hydrolysis. So, lysis is breaking. So, hydrolysis of your ester bond, your peptide bond, your carbon-carbon bond, carbon halide bond. So all this is hydrolysis enzyme. The other one is lyases. So lyases is breaking down. Okay. So lyases is the removal of a particular group. Removal of particular groups. So that is what this group of enzyme catalyzes. So these are your lyases. So suppose if I have, so here it will be like this. Okay, so it will be C double bond C and XY separately. It will remove this group from this. So that is this group of enzymes known as lyases. So you want me to give a recap of biological kingdom? Okay, yes, others, what about you? What is your opinion? So number 5, so you have 6 classification of enzymes. Number 5 is isomerases. So here when you have glucose 6-phosphate, it is converted into fructose 6-phosphate. So here it is isomerases enzyme that plays a role. So the last one is ligases. So this joins 2 groups together. So this is ligases. So this is linking together. So this is classification and nomenclature of enzymes. Clear? So the only last part that I am left to teach here is your cofactors. So when I talk about your enzymes, yes, all enzymes are proteins. Indefinitely, there is no doubt. But your enzymes also have non-protein part. And it also has a protein part. So one example is of your pyruvate dehydrogenase, which we spoke in your link reaction, right? So this is a multi-enzyme complex. So this has 5 subunits. It has 4 cofactors plus 1 metal ion. But this is basically an enzyme. It is a protein, but it has non-protein part as well. For example, when I say Mg2+, it is not a protein, but it is also part of this enzyme, right? So that comes under the non-protein part. Pyruvate dehydrogenase, I'm sorry for the spelling. So the non-protein part of enzyme is known as cofactors and the protein part of the enzyme is known as apoenzyme. Clear everyone? The protein part of the enzyme is known as apoenzyme. Thus the whole enzyme is known as holozyme. Is the sum of cofactors plus your apoenzyme. So please remember this. Your holoenzyme is the whole enzyme, which is your non-protein part plus your protein part. Cofactors plus apoenzyme. Okay? Yes? So plant physiology, recently I have completed, maybe very quickly after, so this is biomolecules. After this I will do cell cycle, cell division, then I will start with your plant physiology. Okay Vaishali, because very recently I have completed plant physiology. So please remember this. So now your cofactors are of 3 types. What are the 3 types of cofactors? So first cofactor is your prosthetic group. So cofactors are basically non-protein part, but your prosthetic groups are tightly attached with your enzyme portion, with the protein portion. They are tightly attached to the apoenzyme. So that is your prosthetic group. So example of your prosthetic group will be your heme. So heme is tightly attached to the enzymatic portion and it is seen in the enzyme catalase or peroxidase. So this converts hydrogen peroxide into water plus oxygen. So here a heme group is also attached. So this is your prosthetic group. Okay? Yes. So number 2, the next one is your coenzymes. The only difference between prosthetic group and coenzyme is that this is transiently attached, or it is attached very shortly with the or to the apoenzyme. It is transiently attached to the apoenzyme. It is not tightly attached. Okay? Yes, clear? So if I talk about the coenzymes, so coenzymes are mostly cofactors, and the basic portion of your cofactors are basically vitamins. Flamin mononucleotide, right? So here basically the essential parts are vitamins. So FMN, FAD, they are all your coenzymes. All right? Yes. The last part of your cofactor, I have no place. The last part of your cofactor is your metal ion. So your metal ion has a unique property. So your metal ion is in the center. So here it attaches to the protein part, that is apoenzyme, and here it attaches to the substrate as well. And this bond is known as your coordination bond. Here also it is coordination bond. Here also it is coordination bond. Okay. So one example will be your zinc. So zinc is used as your metal ion in case of your carboxypeptidase. The enzymes are given in your textbook, so please read your textbook. So this is about your biomolecules. So I am done with your biomolecules in 4 lectures. So please read your NCERT. So tomorrow we'll have discuss questions, but it will be mostly like a quiz. So you will self-evaluate yourself, but I will discuss the questions with you. So tomorrow in Mendelian genetics, we are going to talk about genetic disorder, like what is color blindness, what is hemophilia, how to solve questions based on that. So we are going to discuss, it is at 4 p.m. and again tomorrow at 6 p.m. we are going to discuss important questions from biomolecules. So after this, I am starting cell cycle and cell division. Okay? Yes? So thank you so much for joining my lecture. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So have a great evening ahead. And this is me wishing you most and more. So stay tuned. So tomorrow I'll meet you at 4 p.m. So guys, please do like, share and subscribe once again. Thank you so much from the bottom of my heart, and stay tuned and bye.